that. So at this stage, I would welcome any more questions into the chat box. I'm going to open up the panel for our three key seaweed speakers, Antoine, Martina and Agnes, if they have any particular questions. And of course, if you have any project related questions, I can direct them to Andrew. I suppose I'll start off with one question that I have for you, Martina. We discussed in depth the potential pathogens for seaweed. What would you recommend to seaweed producers or farmers within this region in terms to mitigate the risk of that, or is there anything that can be done to mitigate the risk of getting any of those pathogens?
It's a very tricky question at this point, because really we are at the beginning of understanding what the pathogens are. But I think something that Agnes already mentioned is really having a minimum amount of sterilization in the hatchery stage will definitely help, and also the selection of the material that you use for your seeding. So as clean as possible. Check out for possible symptoms, and really try to use the really healthy looking algae. Mm-hmm. Then I think during the growing stage, really trying to monitor as much as possible by microscopy your life cycle stages, and if there's something weird and unusual looking coming up, trying to communicate it.
Great. Okay. Very useful. Thank you. Agnes, one question that came up there was looking at what is considered high quality seawater. So Martina mentioned we have to mitigate pathogens coming in through the seawater. So what in your terms is high quality seawater, and what I suppose are your processing methodologies for the seawater coming in?
Yeah, so I guess I misunderstood the question a little bit that I've been trying to answer in the chat. But so I mentioned in the talk that we were doing some analysis from the sea farm, and I thought the question was going on that. But with regard to the water coming into the hatchery, I am, well, as Martina says, if we use seaweed material that looks really good, and we try to maybe wipe off the surface and stuff like that, and there are no spots and dots and strange things, then I think with the pathogens, that's a good start. But we have really had large or big problems in the hatchery with getting a lot of growth of especially different types of diatoms. And they, the first thing that they do is that they are much more efficient in taking up nutrients and dividing, so that will be a huge competitor for the small sporophytes developing in the tanks. So that's a huge problem, and you can only get rid of those if you really filter your seawater down to, well, in the new hatchery we are going all the way down to 1 micrometer, and then after that we are using a UV filter. Because it's in the hatchery phase, you have to consider the seaweed as these tiny little organisms that should not be spending their energy on competing with other algae. And bringing other algae into the tanks that are optimized for algal growth, you will really be growing other stuff if you don't take care and use really clean, well-cleaned water. But one thing that can be done if you have the possibility, and that's also why we are placing our hatchery in a location as the one on Suðuroy, is that if your start material with seawater is from a open and clean environment, and you make sure that your intake of seawater comes from a certain depth, then you will take in water from under the photic zone if you can. That means there will be very little algal material in that water, and when you bring that through your filtering system and your UV system, then your seawater should be of a quality that gives you a good chance of hatching good quality material.
Mm-hmm. Okay, That's great, Agnes. I think that answers the question that Enrique had. There's just one more after coming in for you while we're talking about this, and it's about the substrate, which is the ropes. And Laura has asked, is there any opportunity to use natural ropes instead of petroleum-based ropes? Would hemp be a sustainable replacement? Have you looked into choosing a more sustainable option for your substrate?
I guess that's for me, right?
Yeah.
Yeah. I have thought about it. We have been using the cheapest we can get due to budget. But what we are finding is that if we take care to harvest at the right time, which means harvest in the spring, in May, June, then we can kind of take our crop right off the ropes, and the ropes are left really, really clean. They can be coiled and they can be reused. So in that way, the material is quite easy to work with, and we may be considering, not oil-based material in the future, but we haven't really had the economical opportunity to choose better material, and also when you go to the store, when you buy the rope, that's mostly what you get. If we're going to use other material, we'll have to order that in like special orders, and hopefully in the future we will have an economic option to work with other types.
Certainly, that's something that you can pop down on your list as a to-do, or some further research down the line. Hopefully, we can get involved in that. So moving on to the next question I have for Antoine there, who got a coffee delivery. Can we get the same, please?
Come on, we're in Ireland. We drink tea, obviously.
Very good. So the question I had for you was around the biodiversity and ensuring that we keep that. So just scrolling back up here to the question from Enrique. So if I keep reusing the same genotype, I'll end up with a farm with reduced genetic diversity. Isn't it risky? Do you mind just explaining a little bit about biodiversity and the strain selection and how that works?
Yeah, no, those are very important questions, and I think it's important if and when we embark in strain selection and breeding, to keep in mind not to repeat the mistakes we've made in land plants. And what happened in land plants is that due to over-aggressive selection, let's say, we end up with very uniform crops. So basically all the wheat varieties or the maize variety that we're growing, or the bananas varieties that we're growing, are very uniform genetically, which means that they are very efficient for growing in the system in which they're grown, but they are not very resilient at all. So they don't possess enough genetic diversity to tackle new issues arising, because effectively they've been selected in one particular system, so they're very good for that system. But as soon as the system changes, let's say climate change or indeed a pathogen arriving, not having any genetic diversity is certainly a massive issue. And that's why we're losing all the bananas in East Africa, because everything that's grown is clonal, so there's no genetic diversity in there at all. And there's a new aphid that came in about 20 years ago, and none of the cultivars there possess the genetic material or the good alleles that render the cultivars resistant to the disease. So effectively, all the cultivars are susceptible, so all the cultivars are dying to that pest. So it is very important, and I completely agree with that we need to make sure that we maintain a minimum of genetic diversity within the farming systems to allow for more resilience to changing environmental conditions, or indeed the emergence of new pests and pathogens. And the way to do that, and it's true that when I gave the talk, it's obviously a very simplified version of what a breeding system could be, or a selective system could be. But the idea is to use several selected populations and not just one individual. So a way to maintain a minimum genetic diversity within the farm and within the population. So I hope that helps. That answer.
Agnes, you have a question there?
Yeah, so it's for Antoine. So when I've been thinking about selection and stuff like that, so how do we protect the natural populations? So somehow we have to make sure that our crop doesn't go fertile on the ropes, right? Or
Ideally, yes. Yeah. If you want to try to protect the wild populations, you should obviously harvest your farm material before they become reproductive. But realistically, there will be some escape. I think that there's generally no way to ensure that there's no escape of elite cultivars within the population. The only way to do that, and that's why I particularly like Ulva, is that we can grow it onshore, so it makes it much easier for biosafety reasons. As soon as we are growing kelps or anything in open sea, there is a chance that there will be escape of the selected varieties in the wild. Now, the thing is, the selected varieties will be selected for high growth. For example, let's say high growth is what you're interested in, on rope systems. So there is a chance that if those individuals detach, for example, from the rope, they may not do very well when they are in the open sea or in the seafloor, etc. And in that context, that particular sample may not actually spread its genes to the wild population. But there genuinely is no way to protect completely against that outside of growing it onshore.
Yeah.
So, but that's why we'd advise using local populations as a starting material, because at least if there's escape, it's still a local population that is spreading within the area and not.
But the
not
question
something...
is how local should it be? Because from the, for example, so the material that I sent to your lab, we could see the difference in population genetics between just populations in the tiny Faroe Islands. So my principle that we work in the hatchery is whenever we produce lumpfish shelters to one location, we go to that particular location and take the spores. And we will probably continue doing that, but just, maintaining the cultures from those different locations, and then be seeding with different genetic material wherever our ropes are ending up. So it's
how local is local. There's no good answer to that, I'm afraid. But I suppose an objective way of looking at it is whether that genotype, so that individual, could end up in that area naturally, so without human interference. I suppose that's an objective way of defining it, but there's no good answer to that.
Thank you to both there on that interesting topic. There's still a lot to debate there. We just had one question in from you and Scott, more of a question for Andrew. Andrew, you've addressed it there in chat, but just to explain a little bit, we have been looking at waste production of seaweed or seaweed byproducts, so maybe even storm cast seaweed on beaches, and possibly using that in a pellet system for combined heat power system or for gasification. We are currently in the prototype development stage of that pellet. A press release will be out tomorrow with a little bit more detail on that, but that's something that's currently being progressed, and hopefully in the next couple of months, in the next few press releases or towards the end of the project, will be clearer what can be achieved with waste byproduct or with storm cast seaweed in terms of gasification and pelletization there. But thank you all. Thank you to our 4 speakers today for participating, and for the audience. I hope you found it quite a useful seminar. It will be recorded, or it is recorded, and it will be available on the YouTube channel if you'd like to look back or send it to someone else as well. So I'd like to thank you all for taking part today, and hopefully we'll hear from you all again soon.
Thank you for having us.
Thank you.
Bye-bye.